The course we took this week was Pre-Med How Organisms Develop, which includes topics such as cryptobiosis, metamorphosis, in vitro fertilization, cardiogenesis, more specifically the generation of a heart in a chick embryo, and regeneration.
So, as stated before, we did talk about a lot of biological processes in this class, which included cryptobiosis. Cryptobiosis is a state where an animal stops all metabolic activity when put into extreme conditions. In this state, it's basically dead until it finds favorable conditions in an environment. As an example, we looked at tardigrades under a microscope with water, then it took the water away and observed the tardigrades in its cryptobiotic state, also known as a ton.
For this experiment, our class went outside with forceps and petri dishes to collect moss from trees in the ground. When we got back to the lab, we rehydrated the moss and drained it. We examined the water left under a microscope, and with some luck, we found some tardigrades, also known as water bears.
In our second experiment, we looked at Artemia. Artemia are small crustaceans, commonly called sea monkeys. When we first got our Artemia, we had small, dried up eggs. These were because the eggs were in diapause. Diapause occurs when organisms know that conditions are not suitable for life. So the metabolic activity stops and the eggs dry up, and basically the eggs were in hibernation. To bring the eggs out of diapause, we placed them in hypotonic and isotonic solutions of artificial seawater, giving them the right conditions for life.
We observed the hatching of the Artemia. This is known as the first instar. At the time, the Artemia were small, but after several hours, the first metamorphosis occurred. The Artemia grew out of their exoskeleton and produced a new one, resulting in an exponential growth in size.
During our third experiment, we worked with sea urchins to observe the process of fertilization. During our experiment, we injected the urchins with a potassium chloride solution. Doing this caused a chemical reaction which shocked the nerves of the gonads. This caused the urchins to either eject eggs or sperm. This process would reveal the urchin's gender. Male sea urchins would release a white sperm, while the female urchins released eggs that would be a lemony yellow. Once we figured out the gender, we made sure to keep them in separate petri dishes to make sure the eggs didn't get fertilized early.
Once we were ready for the fertilization process, we combined the sperm and the eggs. Doing so triggered a chemical reaction called a chemotaxis. Chemotaxis is the process in which an organism is attracted or repelled from a chemical substance. In our case, the sperm was attracted to the egg, resulting in the fertilization of the egg. Over the course of 3 days, we observed the many stages of sea urchin development, such as the first, second, third, and fourth cleavage, which resulted in the division of cells.
The fourth experiment that we conducted was testing and observing cardiogenesis in chick hearts. We analyzed the effect of stimulants such as nicotine, adrenaline, and caffeine on the hearts in terms of beats. After incubating the eggs, we carefully cracked them into saline solution that was the same temperature of the incubator. Next, we calculated the average BPM of the heart under a dissection microscope. Lastly, we added the stimulants in separate trials onto the chick embryo and measured the change of the BPM. We found that both adrenaline and caffeine raised the BPM of the hearts, while nicotine had no significant visual effect. Afterwards, we would cut the embryo out of the yolk to further examine the heart and blood vessels under a microscope.
With the incubated eggs, we also cracked them on saran wrap that was in a cup over the same saline solution. After that, we put a lid over the cup and put it back in the incubator in order to create an artificial eggshell.
For our fifth and final experiment, we performed a regeneration experiment with the help of planaria, which are flatworms. We put these planaria into petri dishes and cut them in different ways. The reason we cut them in different ways is to see the blastema, which forms after the body realizes a body part is missing.
The way the blastema works is by rearranging the remaining living cells, and then it starts to bubble up around the cut. After that, it starts to regenerate, one of two ways. The first way is epimorphosis, where the planaria makes a small tail and slowly grows it out. The second way is morphallaxis, where the cells reorganize and grow out in the shape of the missing head.